The overall goal of this procedure is to collect unbiased representative data within a limited sample and to precisely analyze the complex three-dimensional structure of the tissue. This is accomplished by first capturing virtual slices of immunohistochemically stained pancreatic sections. Then, the virtual slices are quantified with the IHC virtual slice macro in ImageJ software, and the data is analyzed with scripts written for Mathematica. Next, a 3D reconstruction of the virtual slices is performed using ImageJ. The final step of the procedure is to capture a stack of individual islet images using the slidebook software and to map the image stacks manually in 3 dimensions using Stereo Investigator. Ultimately, precise islet architecture with 3D coordinates for each islet cell can be obtained through a combined method of 3D imaging and manual mapping.
This method can help answer key questions in the field of obesity and diabetes studies, such as how physiological and pathophysiological conditions affect pancreatic beta cell mass, islet distribution, and architecture.
The implications of this technique extend toward diagnosis or therapy of obesity and diabetes mellitus, because a better understanding of the changes in beta cell mass will facilitate the development and evaluation of therapeutic interventions.
Though the methods described in the present study specifically provide insight into the dynamic immunohistochemical analysis of the pancreas, it can also be applied to an array of other organisms and tissues.
Visual demonstration of this method is critical, as the computer-assisted analysis steps are difficult to learn because of the complexity of analyzing such large data sets in 2 and 3 dimensions.